Tesileanu. He is an associate research scientist in the lab of Dmitry Chilkovsky at the Flatiron Institute in New York. And he's going to be talking about biological learning of local motion detectors. TV, the stage is yours.
Okay, hi everyone. Yeah, so this is work done with Alex Genkin from NYU and Mitesh Klofsky at the Flatiron. Let me write this. Okay. So I'm going to talk about how to build neural circuits that learn local motion detectors. And in animals from fly to mouse, we have these motion sensitive cells like these retinal ganglion cells that respond to objects moving in some directions, but not others. These are spike trains, and we see this moving down exhibits no response, moving up exhibits a strong response. And it also only works in a localized area on the retina. And for the fly, we have similar cells that respond to some direction of motion, but not other. And in both of these cases, the mechanism does something like correlate the signals from two nearby points on the retina with some different temporal dynamics. Um, so if we think of this baby Yoda dropping an apple and we have two receptors at 2 points on the apple trajectory, we have two cells, one with a slow response and one with a fast response. And by correlating the slow response from the upper cell with a fast response from the lower cell, we can detect motion that goes downwards. So this sort of mechanism is localized, I guess, in two senses. One is that it detects motion only in this area of the visual field, and another is that the connectivity is local. So this cell doesn't try to sample positions from very far away. So we can actually try to wonder why the detectors are localized. So we might imagine that if motion was global, it might actually be useful to try to pull together information from distant locations on the across the visual field. So a possible answer is that it's an adaptation to the fact that natural motion is not global. Motion is usually different in different places across the visual field because there are objects that are moving and objects are of different sizes. So the thing that we're wondering is whether we can learn motion detectors that are localized, like the ones we see in biology, from an optimization problem that uses moving images that are natural or naturalistic in some sense. And the starting point for our learning procedure um, is to view translation as a rotation in the high dimensional space of pixel intensity. Right, so if you think about an image that's translated, translation doesn't change the contrast in the image. So um, the norm of the image in the sense of the sum of the squares of the intensity values at all the pixels should stay the same. There's some edge effect here, of course. So the pixels at the edge actually can change. So the norm does not stay exactly the same, but it should be a good approximation as long as we can ignore the edges. So we want to think of motion as this sort of trajectory that moves on a high dimensional sphere in the space of pixel intensity. And now the question is, how do we learn that rotation? And of course, translations are of you can translate images by different amounts, right? So depending on the speed at which an object is moving, if you look at two consecutive frames in a video, the amount of motion is going to be different. So we want to somehow capture the essence of this translation, the direction in which it's going, and not worry so much about the amount. So mathematically, we represent that by an infinitesimal generator. And the idea is that if I can take something like a rotation, I can split it into very many small rotations. And the small rotation, if you think about it as a linear transformation, looks like the identity plus some small um, modification of the identity that implements the rotation. So you can think of it as this product of identity plus a generator A, or in the continuous form is the exponential um, some amount of rotation, some angle essentially times the generator. So as an example, if you have a rotation in 2 dimensions, this is the matrix that performs that rotation, and you can think of it as in this exponential form with a generator that's an antisymmetric matrix. And this is in general the case for rotations. The generators are antisymmetric matrices. So what we really want to learn are these generators because those allow us to understand um, rotations or translations by any amount. So how do you do that? Well, whenever you have 2 vectors, you can imagine a rotation that turns one into the other. In this case, I show this big circle, and we can imagine a rotation that goes on this big circle from A1 to A2. But so that's one rotation, it happens to be the shortest rotation, the rotation that requires the smallest angle to perform this transformation. Um, but there are many other rotations depending on the plane in which we want to perform the rotation. So if we want to go along um, a different trajectory that's not a big circle, then we can get many different um, rotations that could do the same transformation. So that means that if we want to learn the particular transformation that converts um, frames at time t1 to frames at time t, we might need to average over many examples because any pair of examples is not going to be able to identify the rotation we're talking about. In a more mathematical language, the sort of rotation we get in this case is a rank 2 matrix, but the sort of rotation that we care about if we're looking at global translation, for example, is a full rank, so rank equal to the total number of pixels in the image, which could be very big. Okay, so now in an actual natural um, environment, we can have several different kinds of transformations. In the simplest case, we can imagine translations in a vertical direction and translation in the horizontal direction. So how do we distinguish that in an unsupervised setting? And a simple approach that seems to work is to just use clustering. And we could use any sort of clustering. So we would cluster these rotations that we're inferring from each pair of frames. But we want to think of this as a possible biological implementation of the learning algorithm. So we're using something developed in Mitya Shklovsky's lab for the past few years, which is called similarity matching. And the basic idea is that um, you want to find the outputs that best match the correlation structure of the input. So if you have 2 inputs that are very similar, like these red arrows here, you want them to be mapped by 2 outputs that are very similar. And 2 inputs are very dissimilar, like the orange ones, should be mapped to outputs that are very dissimilar, like the orange ones on the right. So in our case, the x's, the inputs, are these basic rotations that we learn from every pair of frames. And it turns out, and I don't have time to go over this in detail, but it turns out that these need to be anti-symmetrized outer products. So I take the frame at time t+1 and the frame at time t. I make a matrix by taking the outer product of those 2 vectors, and then I subtract the transpose to get an anti-symmetric matrix because that's what rotation generators are. They are anti-symmetric. So this objective function here doesn't look biologically plausible at all. I'm summing over all pairs of samples, but it turns out that we can actually write it as a very simple um, biological circuit that has Hebbian plasticity at these 2 sets of synapses. And this circuit itself on its own just does PCA. But it also turns out, and I don't have time to go over this in detail, but it turns out that if we impose the constraint that the outputs are non-negative, this circuit is going to perform clustering instead. So now we just can calculate the outer products from consecutive frames, feed them through this circuit, and out come an output that's essentially, the output here is essentially just a projection of the inputs with the matrix W, which is given by the weights in the circuit. So those weights are going to be our detectors, our motion detectors. So we're going to look at a very simple example in one dimension because it's easier to understand. So we're going to start with patterns that are essentially just plateaus like this of different lengths. So these are 6 different patterns. You see the pattern at time t and at t plus 1. So these are just plateaus that move by an amount between 0 and 3 positions in one time step. And then we're going to apply a sort of a filtering, a sort of centered surround filter to obtain um, these wiggles that are moving from one frame to another. And these are the frames that we're going to use for learning. After we run this with about 100,000 samples, these are 6 of the generators that were learned. So the thing that we notice here, remember that W's are the connections um, on, sorry. So these are the connections in the networks. And the things to notice is that, first of all, these are local. They respond only to a small portion of the image. And they're also local in the sense that you only need connections between nearby pixels in the image, right? So all the non-zero entries are localized in a small space here. And I'm sort of running out of time, but this looks like sort of classical models of motion generators. And to make this a little bit more intuitive, we can just make a plot. So for each of the filters, I made a pattern that has a peak kind of around the center of the generator and another peak somewhere where the generator is almost 0. And then I can apply the generator to these patterns, and I see that indeed it moves one of the peaks in one direction or the other, and the other peak stays about fixed. So this shows that what I've done here is I've learned generators that motion generators that apply locally. And yeah, I just said all of this. So I'm going to leave this here, and thank you for your attention.
All right, thank you so much, TV. Um, I actually have a very naive question about how you would take into account like jitters in the motion. Like if let's say there is, you know, like essentially how the retina performs the saccade correction or something like that. Do you have
Mhm.
some a similar type of correction that you apply where you're canceling out the effect of jitters in the movement, so that you're not actually applying some type of an actual transformation term to it?
Right. No, we haven't yet thought about this in detail, and it's it's sort of 2 different ways of looking at it, right? So it might, in some sense, most of the motion observed by the retina might come from this sort of self motion, from the eye moving, um, and it might be that that's the sort of motion we should be focusing on rather than um, the actual motion of objects in the world. So I guess I should say that in this sort of artificial example, we haven't specified where the motion comes from. This could be just from the eye moving rather than from the objects themselves moving. Um, but yeah, we haven't thought in much detail about how those 2 things interact.
Right. And another, I guess, very naive question is, so in this um, your matrix, so all these different inputs are essentially uh, different positions of the moving object, right? Like, so you're taking all of those into account, and then, or essentially you're applying. So all of these different inputs are different objects moving, right? As opposed to different movements of the same object.
Um, I guess in some sense that's right because I'm randomizing the sizes of the objects in each sample too. Yeah, I don't think that makes a lot of difference, at least as long as we have some variability in the size of the object. So one thing that's sort of striking here is that objects always come in pairs, right? There's an edge and another edge where the object ends, but the filters that we find don't necessarily come in pairs, right? So some do have both sides, but some somehow focus on only one side. And I think if you had objects that were always the same size, then you might get filters that always have two kind of peaks. But I think as long as you have some variability in size, it's not going to happen.
Right. All right. Well, thank you so much, TV. That was really great. We're going to
Okay.
move on to our next speaker.